Sir,
Okay.
uh,
Okay, very good
three
afternoon,
madams
my
are
dear.
there, no, sir?
Okay. Okay, madam has joined just now. Okay, madam, hearty welcome, Dr. P.MANJULA madam garu, Assistant Professor, Department of Botany, RBVRR Women's College, Autonomous, Hyderabad. Madam has huge experience in the plant sciences, especially competitive exams regarding of our CPGET 2024. So that's why we are wholeheartedly welcoming you, madam. Please share your screen and start your session. In previous class, madam has clearly explained about the anomalous secondary growth in all types of plants, especially the most important Acheronthus, Boiravia, Bignonia. These are the most important plants. How the cambial ring or cambial activity mm, is impact on the secondary growth or anomalous secondary growth. Madam has clearly explained with beautiful slides. So, madam, please welcome and start your session, madam.
Okay, thank you, sir. So, good
Madam,
afternoon, and
if
also,
possible, madam, if possible, please click on slideshow.
okay. Sir, is it okay?
Yeah, yes, madam, please proceed.
Okay. Good afternoon, one and all. So, today's topic is anomalous secondary growth in monocotyledons. So, in last session, we discussed about the anomalous secondary growth in dicotyledonous stems. Okay. So, generally in monocotyledons, that is, they do not show secondary growth because the cambium is absent in the vascular tissue. So here the cambium is absent. So here secondary growth is absent. But so some exceptional cases are there. In this, the anomalous secondary growth will takes place. So here the monocotyledons generally they normally do not show secondary growth because the absence of vascular cambium. So therefore, the plant body consists of the primary tissue only. So in many palms, however, they develop the thick stem by the activity of the special meristem called as a primary thickening meristem. So it is called as a primary thickening meristem. So this cambium, which originates below the region of the young leaf primordia by periclinal divisions. So in monocotyledon, generally secondary growth, it takes place through the cambium. But here in monocotyledon, that is the cambium is absent. So here the special meristem which is developed at the developed below the region of the young leaf primordia, and it helps in the thickening of the stem. Okay, so here the special meristem, it is called as a primary thickening meristem. So this cambium which originates below the region of the young leaf primordia and by the periclinal division. So this type of secondary growth which is seen in these plants, it is called as a diffuse secondary growth. So another term for this is the diffuse secondary growth. So secondary growth by special type vascular cambium also takes place in some woody lily flora members. Okay, so here the secondary growth which may takes place through a special type of vascular bundle, cambium. So generally in agave, dracaena, yucca, and cordyline. So generally, so these are the exceptional cases. In this anomalous secondary growth is taking place. So this cambium, which develops in the parenchyma outside the primary vascular bundles. So generally, when we observe the structure of the monocot stem, so that is the vascular bundles, they are scattered in the cortex region only. Okay. So the cambium which develops in the parenchyma outside the primary vascular bundles. So unlike the vascular cambium of the dicotyledons, dicots, so this meristem which produces secondary vascular tissue and conjunctive tissue towards the inner side and only little parenchyma towards the outside. So the newly formed secondary vascular bundles which may be ampivasal or collateral. So here what happens? So generally here so generally in monocots, secondary growth is absent, but due to presence of some special vascular cambium. So here this cambium which is develops in the parenchyma cells of the outside the primary vascular bundles. Okay, that is the cells which are present outside the vascular bundles region, they becomes meristematic and they behave as the cambium, and they produce the secondary vascular tissue. and conjunctive tissue towards the inner side. So here generally in normal secondary growth, what happens? So that is they produce during the formation, during the secondary growth, formation of cambial ring, and this cambium which produces secondary xylem towards inner side and secondary phloem towards outside. But here this this cambium which produces second, that is the conjunctive tissue and secondary vascular tissue and conjunctive tissue towards the inner side and only little parenchyma towards the outside. Okay, so this the newly formed secondary vascular bundles, which may be, so here it develops into xylem and phloem. So it may be in the central xylem and on either side phloem, or sometimes in the central phloem and on either side xylem, like that. So here it may be varies, or it may be xylem and phloem otherwise. So it like that. So it produces xylem followed by the phloem, like that. So it develops. So example is anomalous secondary growth in Dracena. So it is the typical example for monocots. So secondary growth, which is usually absent in monocots, since the vascular bundles are closed type. Closed type means that is cambium is absent. So a very few eggplants in monocots which show anomalous secondary growth. Example, Dracena, Yucca, Aloe, and Sansevieria and Agave. So anomalous secondary growth in Dracena stem. Okay, so it belongs to family Dracinaceae or Agaveaceae. So primary structure, that is a young stem, is typical monocot stem, and it contains outer epidermis, and it is single layered, and it is followed by the hypodermis, that is sclerenchymatous hypodermis, and next it is followed by the vascular bundles. So here the vascular bundles are closed type, and they are scattered in the scatterly arranged in the cortex region. So anomalous secondary in the dress. So here the secondary growth. So this is the normal structure. So it contains outer epidermis. Epidermis is single layered, and here next epidermis is followed by the hypodermis. Here the hypodermis is sclerenchymatous. Next one is the vascular bundles. That is vascular bundles are scattered and closed type. So during the secondary growth, which is initiated by the formation of special meristematic zone in the inner cortical region. So that is the cortex region only, inner cortex region. So the cells, some cells becomes meristematic and they act as the cambium. So a multi-layered cambial ring is differentiated from the inner cortical cells, which are lying outside the vascular bundle. So outside the vascular bundle, so the cells, the parenchymatous cells become they becomes meristematic and they act as the cambial ring. So this cambium is formed in the region of the stem which sees its elongation. So this will take place after the primary growth. Okay. So the shape of the cambial cells also varies here. So the activity of this cambium is abnormal. So here the formation of cambium which is taking place from the outer cells of the vascular tissue, and the activity of this cambium is also abnormal. So it cut a large number of cells to the inner side and a few amount of cells to the outside. So the cells which are produced external to the cambium are parenchymatous, and the cells which are produced inner to the cambium are partially parenchymatous and partially vascular in nature. So here what happens? So this cambium which produces outside more amount of cells to the inner side and less amount of cells to the outside. So outer cells are parenchymatous, whereas the inner cells, again it is differentiated into parenchymatous and partially vascular in nature. Okay, parenchymatous cells, they're producing parenchymatous cells outside and inner side, and more amount of parenchyma cells to the inner side and less amount of parenchyma cells to the outside. And along with these parenchyma cells, it also producing the some cells are vascular cells. These vascular cells are also produced. So the parenchymatous cells which are produced internal to the cambium, which develop into lignified conjunctive tissue. So these parenchyma cells becomes lignified and they form the conjunctive tissue. Next one. So here the formation of concentric. So the cells of the inner cambial ring which participate in the formation of secondary vascular bundles. So inner side they're producing the parenchymatous cells and partially vascular cells. So these vascular cells helps in the formation of secondary vascular bundles. So usually the secondary vascular bundles which arise from the single vascular bundle initial cell. So the vascular bundle initial cell which divide into anticlinal divisions to form the two R3 cells. That is again it undergoes anticlinal divisions. Like that, so undergo divisions and produce the secondary tissue. So all these cells then undergo periclinal divisions to form the peripheral cells. So later divisions are irregular to form the mass of cells. So like that, so here so the vascular tissue, the vascular initial that is which are produced inside, they becomes meristematic and they undergoes divisions to produce. They undergo anticlinal divisions and periclinal divisions to give the mass of cells. So the centrally placed cell, which are, so they produce the cells, so the mass of cells. So the centrally placed cells metamorphosis changes into phloem cells and the peripheral cells, which are differentiated into xylem elements. So there's the individual secondary vascular bundle, which consists of centrally placed phloem, which is surrounded by the xylem. So it may be concentric vascular bundle or ampivasal type. That is the central phloem, which is surrounded by the xylem cells. So the cambium then cut parenchyma cells like that. So here this anomalous secondary growth will take place. So a newly formed parenchyma cells, which pushes the vascular bundles toward the central region. So after some time, this cambium again behave abnormally to produce the another vascular ring of vascular bundles. So like that, so this will be continue. So this time the portion of the vascular bundles will change and this process continues and many rings of concentrically arranged vascular bundles are formed. So the last ring of vascular bundles are embedded in a mass of lignified conjunctive tissue. So here the conjunctive tissue which is developed from the inner cells only. The parenchyma cells becomes lignified, which are they become lignified due to the thickenings of this lignin and becomes hard structure. So a cork cambium may also develops. So the cork cambium produces the bark. So this is the internal structure of the Dracaena stem. So it contains outer epidermis. Epidermis is followed by the sclerenchymatous hypodermis. Next, so here these all are the vascular bundles which are scattered in the cortex, okay, cortical cells. So these are the vascular bundle. So here the collateral vascular bundle. So here the sclerenchyma. So here this one is a xylem and here the phloem. So this one is a totally ground tissue, that is the cortex region. So here this one is the cambium. The outer layer behaves as a cambium and it produces the, this side it produces the only parenchymatous cells towards outside and parenchyma and vascular, partially vascular cells towards inner cells. So these parenchyma cells, they become this. Okay, so these are the conjunctive tissues. So they produce the inner side, they produce the parenchyma cells. So these becomes lignified. And these are called as a conjunctive tissue and these are the vascular bundles. So this is the structure of the Dracaena stem. So epidermis. So during secondary, here the formation of the cork will takes place. So this is the cortex and secretory cells. This one is a secondary parenchyma and this one is a cambium. So like this, so it produces the vascular bundles. So this is about the anomalous secondary growth in Dracaena. So next one is the anomalous secondary growth in the storage roots, that is the beetroot. So generally in many dicots, the root function as a, so generally in some roots, that is they function as a storage. So the secondary growth in these storage roots is usually anomalous type. So the secondary thickenings in the beta vulgaris, that is the beetroot, which belongs to Chenopodiaceae. And secondary thickenings in the Ipomia batatas, sweet potato, it belongs to convolvulus. So here anomalous thickenings are seen. So these are the example for the anomalous secondary growth in the roots. Okay, so these are that is anomalous secondary growth in Echinanthus, Borhavia, Bignonia, and Dracaena. So these are the stems. But this beetroot, it is the root. Okay, so it is a storage root. Here also anomalous secondary growth will occurring. So example for anomalous secondary growth in monocots, example is the mainly Dracena and Yucca agave. So secondary growth in the storage root. So generally it is a taproot and it is a typical diarch root. So the beetroot, it is a taproot and it is a typically diarch, that is 2 in number. The vascular bundles, they are arranged. So here in roots generally the xylem alternating with the phloem. So like that 2 xylem and 2 phloem rings are present. So the first ring of cambium, which arises from the pericycle outside the protoxylem and from the conjunctive tissue below the phloem. So here in beetroot, that is the formation of cambium will takes place from the pericycle. Okay, so from the pericycle, which is outside the protoxylem. So protoxylem to so here in roots, that is protoxylem towards exarch condition is seen. So that is protoxylem towards periphery and metaxylem towards center. So here the cell, the cells from the pericycle. So here the formation of the cambium, which takes place from the cells of the pericycle. Okay, so the ring of cambium, which shows abnormal activity, and it forms the patches of parenchyma, which are alternating with the xylem on the inner side, and the outside forms the phloem above the xylem. So here it producing the xylem and parenchyma at the inner side and phloem outside. So thus the ring of conjoint lateral vascular bundle is formed, which are embedded in the parenchyma. So the secondary xylem has few vessels and is mostly made up of the xylem parenchyma, which stores the food materials. So here this cambium, so here mainly first the cambium which arises from the pericycle, the cells of the pericycle becomes meristematic and they form the cambium. So here this cambium which produces this cambium which produces the parenchyma alternating the xylem on the inner side, and outside forms the only phloem. Okay, so it producing this cambium cell which producing the cambium ring, which producing the parenchyma and alternating with the xylem and parenchyma cells towards inner side and phloem to the outside. Okay, so like that. So in this way here it producing. So after some time, this cambium which stops its activity and the pericycle cells which divide periclinally, one of the inner layer of the cell becomes again meristematic and again function as a second cambial ring. So this ring of cambium is also similar to the first one in its activity, and it forms the another ring of vascular bundle. So in this way, so here it produces this cambium which produces parenchyma cells and xylem towards inner side and phloem towards outside. Okay, so like that. So this will continue. So after some time, this cambium also ceases to function, and the second accessory cambium or third ring of cambium will develops outside the vascular bundles from the pericycle cells, and this cambium also adds again another ring of vascular bundles. So subsequent cambial rings are formed in the same manner and forms the concentric rings of vascular bundles, which could be seen even with the naked eye. So the first few inner derivatives of each cambium differentiate to produce the parenchyma and lignified elements, which are formed subsequently. So hence each vascular tissue which is separated from the other by the wide radial zone of parenchyma. So here, so when we observe the TS of the beetroot, so it shows these are the parenchyma cells and they are alternating with the, so these parenchyma cells, they store the food materials. Okay, so it produces, so xylem and phloem, which is separated by the parenchymatous cells here, like this. So it produces, so this here, first one is the cambium, which is developed from the pericycle region. The cells of the pericycle becomes meristematic and forms the cambium. And this cambium which produces parenchyma cells and xylem towards inner side and phloem towards outside. So due to which what happens, the xylem and phloem comes under one side and parenchyma cells are separating these vascular bundles like that. So these parenchyma cells, they store the food materials. So like that, so here they show the alternating rings of vascular bundle separated by the parenchyma cells. So this is about the anomalous secondary growth in beetroot. So any doubts?
Ma'am,
Tell me.
ma'am, this Dracena stem you explained now, ma'am.
Mm.
That diagram must explain, ma'am.
So Dracena stem. So here Dracena, it belongs to the monocotyledon. Monocot.
ma'am.
So generally monocots, the structure of the stem, it shows. So here the vascular bundles are scattered in the cortex region only. Corticals. So here there is no special what you call endodermis, pericycle, or pith medulla, or medulla. So these are scattered in the cortex region only. So each vascular bundle here, this one is each vascular bundle only. Okay, so here during the anomalous secondary growth, what happens? So the cells of the pericycle becomes meristematic and they behave as a cambium, and it producing. So this is the cambium, for example. So this is the cells which are developed from the pericycle region. Okay, so this cambium which producing less amount of parenchyma cells towards outside, and inside they producing the parenchyma cells and vascular cells. Okay, so this parenchyma cells becomes liquefied and form a conjunctive tissue, and this partial cell vascular cells, they produce so they undergoes continuous divisions of anticlinal and periclinal divisions to form the mass of cells. Okay, so the central cells becomes metamorphosed or modified into phloem, and peripheral cells becomes act as a xylem. So that is the condition, that is ampivasal, that is a phloem, surrounded central phloem which is surrounded by the xylem. This condition it is called as a ampivasal vascular bundles. Okay, is it clear now?
Okay, okay, ma'am. And
Okay.
madam,
Tell me.
you said that in Dracena it having that epidermis and hypodermis and vascular bundles, no, ma'am. These three are one is epidermis is outermost, ma'am.
Epidermis outermost,
And hypodermis,
which is
ma'am.
followed by the hypodermis, epidermis, which is followed by the hypodermis. But here what happens? Here the hypodermis is not there in this structure. Your doubt is this?
Okay, ma'am.
So during the secondary growth, what happens? So this here formation of the cork cam, that is cork cambium, that is outer cork formation will be taking place. Okay, this one cortex region only, hypodermis here. This one only hypodermis.
But in diagram they have not labeled that it is having hypodermis, no, ma'am.
Hypodermis is nothing but this cortex only. So the outer layer of the cortex is generally we call it as a hypodermis. Okay, outer. So here the cortex is again differentiated into hypodermis, general cortex, and endodermis. So this total cortex means here comes and hypodermis, general cortex, and so sometimes in this cortex it is differentiated. So outer layer of the cortex it is called as a hypodermis. That is this the layer which is followed by just beneath the epidermis, which is called as a hypodermis. Okay.
Ma'am, then what is about vascular bundles?
Vascular bundles, they are present, they are scattered in the cortex region only here. So these all are the vascular bundles only, they are scattered in the tell me. Hello. Okay. Is it okay?
Okay, ma'am. Okay, ma'am.
Okay. Okay, sir. So I will stop here.
Ma'am,
Tell me.
ma'am, in CPGET examination,
Okay.
it may,
Your voice is breaking.
if suppose writing 2 subjects, ma'am, 2 subjects are given in
Voice is breaking, Naina.
ma'am, in CPGET exam, no, ma'am. Is that
పోయినట్టుంది. సార్ లిఫ్ట్ చేసి కట్ చేస్తా అని చూస్తుంది.
సార్ గుడ్ ఆఫ్టర్నూన్. తిరుపతి సార్. తిరుపతి సార్ గుడ్ ఆఫ్టర్నూన్.
ma'am,
ఐఎమ్ ఆస్కింగ్ టు సార్.
I'm asking that.
Okay, madam,
Okay,
thank you
ma'am,
very
okay.
much. Your session is a very live session and wonderful presentation, madam, with beautiful slides. Thank you very much, madam. So tomorrow, same time, we will going to through Zoom. Thank you very much, madam.
ఓకే సార్, థ్యాంక్ యూ సార్.